Good afternoon. I'm Brian Cholewa. I am a regulatory toxicologist at the National Cancer Institute. Even though I will discuss a little bit about why we're not a regulatory agency and why we don't have a general focus on toxicology, but still the topics I discuss will hopefully be of interest to this group. So my disclaimers is I have nothing to disclose, and these are, my opinions and don't necessarily represent the NCI. And also these are informal topics and not meant to be specific to funding opportunities. So a quick overview, I'm just going to talk about our role in carcinogenicity studies, our focus on drug development, the intersection of the NCI and carcinogenicity studies, and a little bit about a couple of our drug development pipelines. So as I was saying, what we are not a regulatory agency, and we do not have a focus on carcinogenicity testing as it relates to toxicological methods. So even though we have a heavy focus on carcinogenesis, we're one of the leading funding agencies in the world for it. In the context of method development, when we're speaking of replacing a 2-year animal model that we would typically see either in late phase drug development or something that we would see testing pesticides, herbicides, agrochemicals, things of that nature. We do not have a specific focus on that, but I do think we have areas of interest that align. So what we are a funding agency. We have intramural and extramural focus on carcinogenesis as it relates to disease prevention, interception, and therapy. So not so much the toxicology side of it, but how it relates to the actual disease of cancer. And that's primarily what we fund. But again, I do believe that we have a lot of overlap with the method developers, and hopefully you'll agree. So just a little bit of why we don't focus on carcinogenicity testing in this context is, for one, the bulk of our drug development within the NCI is a focus on early phase clinical research. So, typically your phase 0 exploratory, phase 1 safety studies, and your phase 2 preliminary efficacy studies. Anything beyond that, going into the phase 3 and plus, we generally pass off to our industry partners, and we allow them to take those forward. And typically where you're going to see carcinogenicity studies are at the time of going into an NDA, so a new drug application, which is your registration trial going for the FDA. So for approval, for drug approval within the United States. So this is probably a pipeline that everybody is very familiar with, whether you're in the drug development area or not. But this is a typical drug discovery program going from early research and discovery all the way through the review process into where you would typically see FDA approval. So a lot of our focus, as far as toxicology work, goes into this preclinical testing phase. So in this early first several years, and this is a slide from the FDA, and these are the general timelines. It takes about 10 years to get a drug from conception through to approval. And so this first phase generally takes 3 to 5 years, and there's usually quite a bit of toxicology studies done here. So this is when we're going to start doing our initial dose range finding studies, our phototoxicity studies, our mutagenicity studies, so our Ames tests, things of that nature. We'll go into our repeat dose toxicology studies, our GLP studies, which are generally considered our IND enabling studies that will allow us to get into this phase 1 area. We try to make sure that our clinical durations are covered by our non-clinical studies so that we can anticipate any toxicities that are going to be associated with our drugs. But within the NCI, this is about where we stop, is at this phase 2 mark. And so here I've outlined generally where you're going to begin your later stage toxicity studies. So your reprotox and your carcinogenicity studies. And so being in this late phase 3 plus development is why we generally don't focus on these carcinogenicity studies. Okay, so this is going back to us being a funding agency. So in 2022, these are all available, publicly resourced. I did not use any internal information for this presentation. So everything that I'm going to present here is available publicly on our cancer.gov website. And so we have an annual budget of about $7 billion, and 74% of that is an extramural research budget. And while we don't focus on carcinogenesis as a toxicology assay, we do fund a lot of the identification of carcinogenic markers. And this is just a breakdown of where that funding goes in the fiscal year of 2022. So here I brought up a couple examples of 2018. So these were in the funded research available pages of the NCI. And if you look for biological carcinogenesis by keyword, there is a specific grant term that is used for those. There are 0 registered biological carcinogenicity grants that were funded between 2015 and 2018. 2018 was the last year that was fully available on the public-facing webpage. But if you just search for the term biomarkers, you'll see that there's 563 funded studies in 2018 alone. And so here are just a couple examples of the different types of biomarker studies that are available. So you have your CT, so your circulating tumor DNA studies for early detection of monitoring colorectal cancer. And the second study here outlined is the development of early detection biomarkers in small cell lung cancer. And in the second study, these are protein biomarkers. So you can have circulating tumor DNA, you can have protein biomarkers. And I think this is where we have an intersection between the NCI and method developers in this area when we think of toxicology studies, in the sense that if you want to shorten the 2-year typical range that you're looking at for a carcinogenicity study, early biomarkers might help us identify the development of carcinogenesis long before we have palpable tumors or anything noticeable within the animals. This might give us a strong sign for carcinogenesis earlier. This would reduce the cost of the studies, would reduce, the tumor burden on the animals, a lot of different components there that would fit under the 3 Rs. And secondarily, if we are looking to go in vitro, we can imagine a scenario where we're going to have liver-specific toxicities, or even if we go into organ on a chip and human on a chip technologies, we might be able to utilize these biomarkers as a method to predict whether or not an agent or an agrochemical or anything else will be carcinogenic. And in our case at the NCI, we fund a lot of research into these early detection biomarkers, whether they're diagnostic or prognostic. So these might be valuable tools to anybody in this area. And so one of the programs that works on this within my division is the Early Detection Research Network. So this is the EDRN. And this is just a cartoon showing the basic pipeline of, you have discovery, assay development, validation, and this is going into clinical validation, which leads back to discovery of new potential biomarkers. And my case here would be that we can take these early detection biomarkers and utilize these in these carcinogenic models, and we would assume that some of the markers would be the same, whether the cancer is developed naturally through, which is what we generally are looking for, is the natural development of these cancers so that we can target them early with therapeutics, or if we're actually inducing these cancers ourselves through some sort of environmental exposure. And so out of this program, these are the FDA-approved tests that have come out. Just to highlight some of them, there are additional ones, but here you have, a cancer-seq, so the detection of genetic mutations associated with pancreatic and ovarian cancers. So these are genetic mutations we might be able to look for beyond the ctDNA and the proteins. And then, here is this is a panel of biomarkers for the assessment of hepatocellular carcinomas. So again, this comes back down to a panel of biomarkers that may be utilized in these toxicology studies that might be predictive of carcinogenicity. These are, I think, again, valuable in cross sections where we might be able to collaborate and work together. And speaking of that, so how can we collaborate? Well, there's numerous different ways. So we have contract mechanisms such as PREVENT, which is our prevention drug pipeline, and NEXT, which is the experimental therapeutics pipeline. With very similar programs, NEXT is much larger than PREVENT. And PREVENT was modeled after the NEXT program, but these are both our drug development programs. They work on contracts. And within the PREVENT program, this is something that I have a strong interest in, and I'll go a little bit more into that in probably the last slide. But I do think that we can leverage the use of what we're currently doing in drug development with the production of NAMs and create confidence in the NAMs that are being produced and that we test, and also start gaining some regulatory acceptance, particularly with the FDA, which is our primary regulatory agency. But if the FDA is to come on board, then we could assume that other regulatory agencies would follow suit, or at least be more comfortable with, the processes involved. But we also have numerous grant mechanisms. I just listed a couple here. So our R01s, our R21s, UG3s, so the biphasic grants, and anybody is eligible for these. Again, there hasn't been much funding for the biological carcinogenesis models, but at the same time, there's been a lot of funding for biomarkers. If this is something that you're working on developing, I'm sure you have a very strong interest in these biomarkers, and there is funding mechanisms for that, and those can all be found on the cancer.gov website. The other programs that we have, the SBR and STTR, so these are the small business loans, and both mechanisms are similar. If anybody attended AACR in this last week, which would have been April of 24, you would have seen Monique Pond's presentations on these mechanisms. They're great. They're recorded. I would recommend anybody to go in and look if you're in a small business. They have great funding mechanisms there. And the SBIR mechanism is something that I'm actually pursuing. I am trying to get it approved through that mechanism to produce NAMs and utilize them alongside with my preclinical packages now that are being used for drug development. It has not been funded yet. I do hope to get it funded in the next year. And that'll be an opportunity for small businesses to come in with tox assays and applications that would be able to work alongside and utilize the data and knowledge that we have within our clinical trials program. And also we have CRADAs, so these cooperative research and development agreements. These can be partnerships between any federal and non-federal agency. And this really just allows us to collaborate, work together, and as opposed to a funding mechanisms, these are just generally collaborative agreements. So those are several different ways that we can collaborate. So this is the next pipeline. So this is the NCI's experimental therapeutics pipeline. And this is just one of the drug development programs. I'll also discuss the PREVENT program a little bit in the next slide. But in general, you can just see it goes from early discovery all the way through clinical development. And there are numerous phase 1 and phase 2 clinical trials underway. But if you actually look, oh, excuse me, I went back one. And if you actually look at the phase 3 clinical trials, there are none listed. There are no active listed trials on the website at this moment. And that just goes back to what I was saying. We generally focus on the early phase 0 to 2 clinical trials, and they're generally passed off into, pharmaceutical groups and industry to move for the phase 3 plus trials. And so this is the very similar program. This is our DCP, so the Division of Cancer Prevention Discovery Pipeline. Here, the primary places that I work are in PREVENT and CPCT-NET. So PREVENT is our drug development program where you can come in with ideas, and concepts, preferably with a reasonable amount of preclinical data showing efficacy. And then those can go straight into our clinical trials program. We'll do all of the regulatory toxicology testing. So your repeat dose tox studies, your mutagenicity studies, phototox, the various components that generally go into your IND enabling package. And so we will fund those. They are conducted through our contractors. And then once we have all of that, we can pass that data back to you. We can develop your IND so that you can take it into a clinical trial, or it can go through our clinical trial program, which is the CPCT net, which is again generally phase 0 to 2. And so the reason I bring this up, and this may be difficult with carcinogenesis, but just more broadly in the scope of NAMs, I think this is a very unique opportunity for method developers. And the opportunity here is that we can introduce NAMs into our already being executed IND enabling toxicology package. So what this allows us to do is submit these NAMs to regulatory agencies, which can be used for weight of evidence approaches to safety decisions. So in light of the normal toxicology package or traditional toxicology package that we would conduct, we would add the NABs in there. We would be able to compare the traditional toxicology testing with human data and the NAMs. So we would get our multiple species, typically one rodent, one non-rodent. We would get the NAMs data, and then following the clinical trial, we would actually have human data so that we would have comparator arms between all of those different species and methods, and that can create a lot of confidence in the NAMs that are being produced. And one other thing that we are capable of doing is we have a lot of this data already. We've done a lot of toxicology work, and we have that data available with a lot of known drugs, and we have clinical data available as well. So we know what kind of adverse events and toxicities people have already seen. That is all within our database. And so we can also think about developing NAMs and then putting those back into using the drugs that we've already performed these on, bringing those back, doing the NAM studies with those drugs, and then having all of those comparator arms. So I think that's another great place. Again, this is a little probably outside of the scope of carcinogenicity because there's nothing that we're going to bring within particularly a prevention trial that we would expect to cause carcinogenesis. That is never an endpoint that we expect to see. But in general, these biomarkers and things of that nature are available to us. And so I do think it's a very unique opportunity for us to collaborate with method developers. So in conclusion, this will be my last slide, is just, while our focus is not on carcinogenicity toxicology models, we have very similar objectives and are excited to pursue any potential collaborations. And please feel free to contact me. My email is listed here. It's just my name at nih.gov. And all programs that I discuss and the program staff that are associated with those are available on our website, cancer.gov. And I'd be happy to answer any questions via email or just reaching out directly. Thank you very much.